all, welcome to the MOOC lecture series, an initiative taken by LJ Institute of Pharmacy. In this session, given by myself, Tosha Pandya, Assistant Professor, Department of Pharmaceutical Technology, we will be talking about biosensors. So let's begin with the session. Today we are going to see about biosensors. What are biosensors? So they are analytical device containing immobilized biological material, which may be an enzyme, it can be nucleic acid, hormone, or even whole cell, which can specifically interact with an analyte and produce a signal, which can be physical, chemical, or electric signal that can be measured. Now, an analyte may be glucose or urea, drug, or pesticides whose concentration needs to be measured. Okay, so in if you divide the definition biosensor, bio means any biological material which is immobilized and its concentration is measured with the help of a sensor. So let's see the components of a biosensor. In the biosensor, you have a sensor which is sensitive biological element, which may be tissue, organelle, cell receptors, etc. Then you have a transducer which will convert the signal from one form to another. And normally in the biosensors, the biochemical activity is converted to an electric signal. You have amplifier because the signal which is generated is very small. So you require an amplifier to amplify the signal large enough to be measured. Then you have a processor which will process the signal, and finally you have the display unit on which the signal will be displayed. So here you can see the components of the biosensor. You have analyte which will interact specifically with the bioreceptor, which may be enzyme, antibody, microorganism, or the cell. And you have a transducer which will convert signal from one form to another based on different principles, based on the pH change, heat, light, or mass change, etc. And the signal is finally sent to the detector. So basically the working principle, they are operated on the principle of signal transduction. The bioreceptor is allowed to interact with the specific analyte, and the transducer measures the interaction, outputs the signal. Simple, okay? The intensity of the signal output is directly proportional to the concentration of analyte. Means higher the signal output, that means the higher the concentration of the analyte. Now, what should be the main features of biosensors? They should be highly specific for the analyte. The reaction use should not be affected by any factors like pH, temperature, stirring, etc. The response should be linear over some range of the analyte. The device should be tiny and biocompatible. Okay, it should be compact enough to be carried and portable. Also, the device should be cheap enough so that many people can afford it, easy to use, and capable of repeated use. So you have different types of biosensor depending on the principle used to analyze the signal. So first one, you have the calorimetric biosensors, which are based on the heat generated during the reaction, exothermic reaction. Example is the use of glucose oxidase for determination of glucose. Then you have potentiometric biosensors, in which you have electrodes which convert the biological reaction to the electric signal. Example is the urea biosensors are example of this type of biosensor. The acoustic wave biosensor mainly uses piezoelectric materials like the quartz crystal to generate acoustic waves, and they are used coated with usually antibodies which bind to complementary antigen. Then you have amperometric biosensors, which function by production of current when a potential is applied between two electrodes. And the best example is the redox reactions. And the optical biosensor, as the name suggests, it is based on some light which is generated during the reaction, like the luminescence generated using Filey enzyme, luciferase for detection of bacteria in the food. Now, what are the applications of biosensors? So you have seen number of properties of biosensors which are very useful detecting the sample. So they are finding wide use in industries like food analysis, biomolecules, drug development, crime detection, forensic, then medical diagnosis, environmental monitoring, industrial process, monitoring glucose level in diabetes, wastewater management, and agriculture industry. So let's see each of these applications in detail. So how they are useful in food industry? Now the deterioration by E. coli is a major problem with the food products. Now this E. coli can be detected in the E. coli contamination can be detected in the food with the use of urease for by detecting the pH change caused by ammonia produced by urease E. coli antibody conjugate. okay, which is very good application. We can easily know whether the food is contaminated with E. coli or not. The enzymatic biosensor also employed in the dairy industry. Then the biosensors in medical field. In the medical field, as widely applicable, the determination of glucose measurement with the help of glucometers are a boon to diabetic patients, which we can measure portably. We can carry easily the glucometers and determine the amount of glucose in your blood. Then, apart from that, you have hafnium oxide, which is used for detection of human interleukin, early stage detection of human interleukin. In drug discovery and drug analysis, also biosensors find application. They also, the best use also is in the environmental monitoring, in which you can find the herbicide pollution in water or pesticides pollution can also be detected with the help of biosensors. The concentration of ammonia can be defined with microbe biosensor using nitrosomonas species as the immobilized enzyme. The epigenetics is a new branch which detects the tumor cell in a urine sample to an ultra sensitivity level. So here you can see the summing up of the biosensors, the use in pharmaceutical industry. It is a biosensors are completely a boon from the biotechnology. So I hope you are clear with the with the biosensor principle and the working. Thank you all. Please do like, share, and subscribe our YouTube channel Pharma Ignite, and do not forget to press the bell icon to get the regular notifications so that you do not miss out any of the topics in this chapter. Thank you.